Hello friends, welcome you in Research Needs. In this video today we are going to see Actophotometer. So it is used for assessment of CNS activity, and the drug which are showing stimulant and depressant activity we can screen by using this instrument. Now we will see next. Introductions. Actophotometer measures locomotor activity. So what type of activities animals are showing after giving the drug we can see by using these equipments. Indicate CNS stimulant and depressant. So two type of activities showing CNS stimulant and depressant. Stimulant increases activity and depressant decrease the activities. Here you can see the rat which is standing, it's showing activeness, and the animals which is down, it is showing like it is tired or depressant in action. So likewise, while the animals moving active, it is moving, and that this instrument detects. So now principle we will see. Photoelectric cells detect movements, and the movements break lights beams and counts activity. Activity count is proportional to CNS activity. So here you can see it has a light source. This light source produces beam, and that beam is horizontally throws in opposite direction. Here this side, and here you will find the beam detector or receivers. So in between this beam the animal moves. If the animal moves, this light beam breaks from here to here, and it breaks and it is parallel to the floor. So the light beam is break, and as the number of beam is break, it will count. The counter will count the number of light beams are break. So now instrument description. In enclosed chamber with infrared sensor. So here it has you can see the infrared light sources. It is parallel to the floor area. Now digital activity counter, it is a digital activity counter meter. So the if light is break, it will count, and here you will find a reset button. It will make it 0 after activities. Beam breaks equal 1 activity. If one beam will break, it will count 1. Used mainly in rodents. So rat and mouse generally used to evaluate by this instrument. Here you will find a regulator for voltage. It is used to calibrate this instrument. So now we will see how it is used. Use of voltage in actophotometer. Voltage determines sensitivity of infrared beam detections. Lower voltage, lower beam intensity, reduce positive results. So by using this voltage we can calibrate the activity. If animals we have kept in this area, and the voltage increase, and it is after animal movement it is counting, then we can easily calculate that on given voltage the instrument is showing counting, means you can easily use this for the calibration. So next, here lower voltage range. For mice, typically lower voltage range due to the smaller body size. For rat, slightly higher voltage needed to ensure the beam interruption. So first step is to calibrate instrument using voltage regulator in presence of animals. The activity meter is showing counting or not. Used to calibrate the device. So that regulator, voltage regulator used to calibrate device based on species, activity level and experiment needs. The objectives of this instrument is to evaluate CNS stimulant and depressant activities, and that then we have to compare the locomotor activity before and after drug administration. So generally for stimulant we are using caffeine, and for depression we are using diazepam. So here material requirements for actophotometer study, we are using rodents, caffeine or diazepam, serine, weighing valence. These are the material we want. So now we have to calculate dose. How we can calculate the dose? So we have ready animals and we have ready instruments. So first of all we have to calculate the dose. So how we can calculate that? Suppose we have one ampoule that contains 10 mg per 2 ml diazepam. So from this dose is used for human. So from human dose we have to calculate the rat dose. For that the formula is rat dose is equal to human dose into conversion factor. So now after 10 mg divided by 60, whatever will be the mg, that we have to write here, it is 0.17 mg into conversion factor for rat is 6.17. So we will the result will be 1.0489 mg equivalent to 1 mg per kg. So this will be the rate dose for 1 kg rat. 1 mg will be the dose. Now maximum dose volume, now we have to calculate the dose volume. So maximum rat dose volume is 10 mg per kg as per the OECD guidelines. So 10 mg per kg. So from this we can calculate the stock solution. How? Stock solution means rat dose divided by dose volume. So from this formula we can calculate stock solution. So desired stock solution will be 1 means 1 mg divided by 10 equal to 0.1 mg per ml will be the stock solution we have to prepare. So this is the formula by which we can calculate stock solution for animal study. Now we will go ahead. Animal dose, suppose we have animal and that its body weight is 135 gram. So here for 1 kg rat 1 mg dose, here we have calculated that. So we have converted the kg in gram. So 1000 gram is equal to 1 mg dose we needed. So for 135 gram rat, how much dose we needed? 0.135. By cross multiplication you can calculate this, 0.135 mg. So here 0.1 mg in 1 ml. So here stock solution. So here 0.1 mg in 1 ml. So 0.135 mg in how much ml? It is 1.35 ml. So this will be the dose for this gram of animals we have to give. So how we can prepare the now stock solution? This concentration we have decided here. Now we have to prepare that, the stock solution. So how we will calculate? We have to prepare this stock solution 10 ml. So what we will do, in 1 ml 1.1 mg is, so for 10 ml how much it will be? 1 mg in 10 ml. For that our ampoule is contained 10 mg in 2 ml. Therefore we will take 1 mg from that 10 mg. For that we have to divide it by 10. So therefore 0.2 ml contain 1 mg. That we will use 1 mg and we will add in 10 ml of stock solution. Now this stock solution is ready. From this 1.35 ml we will use to give the dose for 135 gram animals. So this is how you can calculate diazepam dose By using this video, you can calculate any drug animal dose in 4 basic steps. I have given link in the description. Please watch it. So now experiment procedure. After calibration of the instrument, what we will do? We will record for all animals baseline. Suppose we have 10 animals, so we have to take their readings for 5 minutes baseline, and after taking baseline we will randomize the animals and divide in 2 groups. One group will be the control, and in which we will treat with saline, and second group will be the drug treated groups. That group we will treat with diazepam or caffeine. After 30 minutes we will evaluate each animal's activity in actophotometer for 5 minutes. And lastly, we will compare the control and drug treated groups results. In observations, control minimum change activities, and in control group you will find normal reading. Suppose you have given saline to the animals for 5 minutes, it is showing 150 activity count. In caffeine it will show above 200, and in diazepam the activity count will be reduced around 70 to 80. So this is how you can observe. This type of observation you can find. Now interpretation: increase activity, CNS stimulant. If drug activities is increasing, then the baseline it is showing CNS stimulant activity. If activities is decreasing, then the baseline it is showing CNS depressant activity. Locomotor activity directly reflects CNS drug effect. So application: what is the application of actophotometer? Screening CNS stimulant drug, depressant or sedative drugs. You can also evaluate the anxiety drugs, and for behavioral pharmacology research, and toxicology and neuropharmacology studies. Other purpose for actophotometer is studying anxiety and exploratory behaviors, assessing motor coordination deficient, and measuring fatigue and endurance in rodents, evaluating locomotor toxicity of chemicals, and monitoring activity rhythm in chronobiology studies. So sometimes what happen, you have given any dose, toxic dose, that time animals which are comes in morbid conditions, they are not showing activity and they are silently sit. So that time you can keep the animals in the actophotometers, and if they are showing some activities, so you can easily evaluate that animals is showing activity comparing normal one, and if it is not showing much activities, you can evaluate that it is in morbid condition. So now conclusion: actophotometer is a simple, reliable tool for CNS drug testing. It is easily differentiate stimulant and depressant activity of the drug, and also widely used in behavioral and toxicological experiments. Thank you for watching Research Needs. Thank you very much.